Okay, thank you everyone for joining us this evening. It's a great pleasure for me to introduce this professorial lecture from Professor Vikki Rand. Vikki joined us in 2019 as a professor and in December 2021 was made interim director of the New Horizons Centre. Previous to that, Vikki started her career at the University of Bradford, where she got a first-class degree in biomedical science and then studied for a PhD in genomics and bioinformatics at Cambridge University. Subsequent to that, Vikki then had an illustrious career as a postdoctoral researcher. She worked at the Johns Hopkins University, the University of Nottingham, and then for 10 years was working at Newcastle University. 7 of those years were as the Bloodwise Senior Bennett Fellow. It's a great pleasure to introduce Vikki's research, which has focused very much on the biology of lymphoma and other cancers, and the main thrust of which has really been to seek to develop new treatments and strategies of treatment. Throughout her career, she has undertaken various research projects focusing on those areas and the genetics that underline them. The title of her inaugural lecture is Geordie Genes and the Power of Sequencing. So without further ado, I'll hand over to Vikki and look forward to hearing her lecture. Thanks, Vikki.
Okay, thank you very much, Steve, and thank you everybody else for attending and also online. So I'm going to talk to you about Geordie Genes and the Power of Sequencing, and my aim at the end of this is to get you as excited about genomics and the future of genomics at Teesside as I am. So just to start off with the overview of what I'm presenting about, we're going through landmarks in genomics. I'll be talking about the Human Genome Project. I'll be talking about the research I've been doing in brain cancers, also in blood cancers, and what's happening at the National Horizons Centre, and basically what the future holds for us all of us. So there isn't going to be an exam at the end of this one, so you're okay, you can all relax. Don't switch off though, student lecture away. So basically landmarks in genomics, there's a lot of exciting things happened over the course of many years. So we're going to focus on just some of the really exciting highlights, and I'll also interlink a few personal bits along the way. So 1953, discovery of the structure of DNA in the double helix. You probably have heard of this and various celebrations, and I think there'll be another celebration next year, 2023, which I think we should definitely be doing some exciting things for. So basically, Rosalind Franklin at King's College London, she obtained an image of DNA using X-ray crystallography. This was an idea first approached by Maurice Wilkins, who was her supervisor, and basically a striking manner of features characteristics of helical structure. And you can see from this is like they're basically looking down on DNA. Also, at the same time, this image allowed then James Watson and Francis Crick to create their famous two-strand or double helix model. And it's quite an interesting papers to read because the way they're written is very different to how we write papers nowadays. I mean, it's basically written as a letter to the editor. And as you can see on the bottom of this one, we wish to suggest a structure for the salt of deoxyribose nucleic acid. This structure has novel features which are of considerable biological interest, which I think is probably a major understatement. So basically Sanger sequencing, so 1977, kind of the peak, this is the first DNA sequencing method developed. So this is how we can take that double helix and kind of pull it apart and understand kind of what's happening. This was a chain termination method, and again, there's no exam, so don't worry about the detail. Where basically a DNA segment is replicated over and over again. And we put in a particular chemical which can randomly stop the creation of that DNA at any point at each of those 4 bases. So basically you take the sample and you split it into 4 and you run it down 4 lanes. So G, A, T, C are the 4 main letters of DNA. So this basically is then superseded by fluorescence technology. So originally it was done with radioactivity, which a few people have told me how they did that in the labs originally, and I definitely would not pass health and safety nowadays. So moving on to kind of the fluorescence, we end up with big fingerprints like this, where we just go down, this is a single strand, a part of DNA that we're sequencing, and we pull out the different colours, and then each one of those represents a different base, and this is how it was done. And I'll talk a bit more about that detail. And although this technology has been superseded by next generation sequencing and other technologies, we still use traditional Sanger sequencing for a lot of the validation. So it's still a technology which is used nowadays. And also, this also coincides with the year of my birth. So thanks to my mum for finding my wristband and various other bits to conclude in this talk. And now my lab can calculate my age, as we were discussing earlier. So also in 1985, another key event, so PCR was invented. This you will have heard a lot about recently with, I don't want to mention the C word, but the C word. And I think it's a really powerful tool. In 1985, it first invented and published. This coincided with me moving from Devon to the northeast. And we've got the train down through Devon. This is Dawlish, and you may have had the sea fret or waves hit the side of the train. Moved up to Burnmoor, where my Geordie genes were returned. So this is where my grandparents were married, and all my family are from the northeast region. So other key points, so 1990, so the Human Genome Project begins. This is the next part of the talk. So 1990 to 2003, what happened amongst those. And 1991 was the first genome centre was established. And what you can see from this picture here is there isn't just one genome sequence, there's lots of different maps. So how the genome was made was, well, the sequence was basically, they used multiple different ways of looking at chromosomes and DNA. So looking for markers to try to basically anchor the sequence in. So it wasn't just a case of we take that technology and we sequence. There's a lot of foundations and infrastructure and kind of proposed first. And I'll show you a little bit about how I used that during my PhD. And this coincided me doing my GCSEs and loving biology at Parkview in Chesley Street, so not too far away. So in 1997, this was the first, E. coli genome was sequenced. E. coli has 4,000 genes, and it was quite a significant paper in terms of a complete genome. Also coincided with me working at GlaxoSmithKline at the New Frontiers Science Park, which is no longer GlaxoSmithKline, they moved back to Stevenage. And I was growing a lot of E. coli in giant fermenters. So this links nicely with a lot of the work I'm doing now with Fujifilm and bioprocessing and manufacturing. Where I basically had a love of medical microbiology, and what I realised it was the love of genetics that underpinned it all, and that manipulation. So this is where I go out during my undergrad degree. It also where I discovered genomics. So one day I was allowed into the new GSK genomics suite, and it weren't all playing solitaire, but I kind of feel in the late 90s we may have been using computers mostly for playing solitaire. But this is where they had, why look at one protein or one gene or one thing at a time when actually you can look at everything. And they were looking at different genes and whatever data was kind of being generated at the time. And that was where my head was turned, and I had to end up here. So the Wellcome Trust Sanger Institute, I joined there in 1999, and the Wellcome Trust Genome Campus is an absolutely beautiful campus if you ever get a chance to visit it. This is the main kind of building area, so this is kind of where my lab used to be. And this is where we used to have our lunch, and this is where we had lectures but also watched the World Cup, and then had a barbecue outside here, and then went back to the lab in the afternoon. So it was a really fun campus and a very, very amazing campus to work on. And there was only one college at Cambridge, very different from Bradford University. So I looked through the different colleges, Gonville and Keys. It had a DNA double helix as a stained glass window. So there was only one place I can go. So Francis Crick was an honorary fellow there. And if you ever go to Cambridge, this is the one next to the Senate House and next to King's College, which actually worked out really well for graduation because they parade you through the town, but this was just round the corner, so it was fantastic. Also, I entered the world of Harry Potter. So this is the main hallway, and I also lived in the fantastic house at the back of King's across the road. And when I opened this book about Stephen Hawking, basically that's my front door, and that's the Duke of Edinburgh stood next to my front door. So a few years apart, that was 1981. So I would have probably been in Carlisle still then. But it's amazing to actually live in Stephen Hawking's old house, and sadly it was demolished before they used it in the film, but that's kind of where it was in the film. I also got asked strange questions, and they use words like matriculation. So I got asked, where did I summer? Previous years I summered at the local pub where I worked behind the bar. This year I worked at Merck Sharp and Dome, basically sat behind a curtain in the dark looking down a microscope, doing some pretty cool science looking at mitochondria activity in cell lines. So basically, how do we kill a cell and then how do we save that cell? And this was my first publication, which is that's how I spent my summer. So get on to the Human Genome Project. So 1999, the full-scale human genome sequencing begins. So the way it was set up was very much about a factory. They took the process from Sanger sequencing, applied biosystems were the first to commercialise it and make it into an automated machine. And then they basically just made it into a factory. So each chromosome had a team. At the Sanger Institute, we did like chromosome 6, chromosome 9, 22, and X, and I'm sure I've forgotten one. But I was part of Team 50, which was chromosome 6. And unfortunately, we have a fantastic giant wooden chromosome 6 photo, and I can't find it with all of us on, where we then used had people who worked in the lab, with different parts of it, who did various bits of sequencing, and then we sequenced them as part of this. And it was just amazing to see. And actually now when you go, it's basically just the next generation of sequencer, which is just fills up the whole area. So we also had the research labs working alongside the sequencing teams addressing key questions. But what you can see from the graph is it's quite a slow start. And it wasn't until kind of around about this time where things seem to ramp up. So the reason it's called unfinished and finished is the Human Genome Project. Like I said, it had those different maps and different layers. So they basically did mapping project, and that was, can we get the frame of the DNA and of the human genome, and then that's how we overlay the sequence on top of that. So we identify the most efficient way of sequencing the genome rather than doing everything. You kind of just do the most efficient path. So the public efforts were sequencing and finishing it, closing gaps, making sure it was nice and tidy, and then doing the next bit. However, in 1999, which just so coincided with the start of my PhD, it just went through the roof. So we'll come on to that. So basically, in terms of my PhD, I was part of chromosome 6, but I was also part of chromosome 9 team. And I was focusing on the MHC region, which is the major histocompatibility complex region, which is a mouthful, but this was the most sequenced region at the time. Only 3% of the genome was sequenced when I started my PhD. What we want to look at was kind of how this compares with other bits of chromosomes. So my job was to sort out a little bit of chromosome 9. So it was this little bit towards the end. So you basically have the ABO gene there, which is the blood type gene, all the way down to what we call the telomere here. And this is what it looked like when I got, part of the way through, when I kind of identified there was bits of sequence here, but there were also gaps. And I could also map different genes into different regions. And a lot of my work was trying to work out kind of how to plug these gaps and also which direction they're supposed to be in. And it's amazing how bits of DNA end up in the wrong place. So how the genome was sequenced, the shotgun sequencing is basically brought down into tiny little pieces. So we have the genomic DNA, which is a bit like having a ball of wool or a big string of beads in one big lump. So what we wanted to do was just chop it into tiny bits and then map it back on and get that most efficient area. But what it looks like down a microscope, which is pretty cool, this is fiberfish. We could take 2 of those little bits here and colour them in different dyes and then work out kind of how close by they were. So by doing this mapping here, I could kind of estimate which bit of DNA was there, which bit of DNA was there, colour code them. Sometimes it didn't go quite so well and it was just nothing because they were nowhere near each other. But here I could actually, this is a string of DNA, the fibre, and this bit is coloured in red and this bit in green, and I could estimate that this is about 100,000 bases long, which meant that there was another little bit of DNA in that library, because they're average about 200,000, that I should need to find to kind of cover that gap, and then that would be classed as finished. And this work was published as part of the chromosome 9 paper, and it's exciting to see your PhD in actual paragraph about my work, and there is a table as well. But actually having that from your PhD was really exciting to see. So the whole purpose of looking at chromosome 9 was because of genome paralogy. So paralogous genes, basically genes that look very similar to each other and they possibly have a common ancestor. So basically our genome went through a couple of rounds of replication. So we end up with bits of DNA that look very similar. There's a lot of things happen in between that, because it's like 400 million years in between. But it's amazing how much evidence there is and why certain genes have been conserved for use. So this is, one gene family, the RXRs. So this is chromosome 1, chromosome 6, and chromosome 9. So my task, we knew this one was there, my task was to find these ones in the genome. And you can do quite cool things because the genome has lots of patterns. So the start and ends of genes, you can actually take them from bases and give them a code. So this is actually a gene structure based on where the stops and starts are in the actual DNA. But also we can look at the different levels. This is the protein, so the DNA makes the RNA, which makes the protein. And at the protein level, you can see just how identical they are. So where there's a star, there's similarities, but also you kind of see where there's a bit at the beginning that they're different, but these key points, they're very, very similar. And I spent a lot of my time doing that. And this was part of a Nature Review Genetics paper that I was part of, and I also got a poster with this on, so if anybody wants, you can still get hold of copies of them. But this basically shows a summary of where all those genes were and how they link back to that original region on chromosome 6. However, the real reason why the sequencing ramped up, it wasn't me, after all. It was Craig Venter. So basically he started Genome Wars. So Craig Venter, he was part of the public effort in sequencing, but he formed a private company called Celera Genomics to sequence the genome because he thought the public effort was taking too long. And it's true. Basically, if that's the speed we were going, if we continued, we'd probably still be sequencing it now. So it was also about new technology bringing in and being capable of doing this. So basically he said, right, we're going to sequence the entire human genome by 2001, but the fundamental part was we're going to make it information only available to paying customers. So that goes against the public effort, who had done I think it was the Bermuda Agreement, I'm not quite sure who organised that meeting, to discuss the ethics and what happens to the data. And they'd agreed, so the public effort, they ended up with 20 sequencing centres from across the globe, and the main leaders agreeing that anything that comes off a sequencer is freely available within 24 hours, and anybody can search that. So it sparked a huge rivalry with the public Human Genome Project effort, and basically who's to be the first to sequence the human genome. But as I said, it basically really started to push costs of sequencing down and developing the new technology. But who won? I would say the sequencing company may. I think Applied Biosystem may have won that one. But it was declared a truce in 2001, and the data is available to everyone. And I'm happy to bore you to tears by showing you how to search for different genes in Ensemble in multiple different species at any point. So the result was this joint announcement that both sides had completed their own working draft of the human genome. And then the same day there was a publication in Nature and a publication in Science. The public effort was in Nature, and the private effort was put into Science, and obviously the genome was available. But they also meant that they had two different genomes. So as a PhD student, it was quite difficult because you had multiple genomes to search to identify these different genes. However, now it's become much more aligned and there is only one kind of reference genome. It also was very political. So there was lots of discussions with Congress and trying to stop things, because obviously with the public effort and the private effort, they could have just pulled all that public funding, but thankfully not. The Wellcome Trust invested a lot more money into the campus and the sequencing, and same with the US as well. So basically you've got John Sulston, who was the director of the Sanger Institute, and Tony Blair, and then in America you had Craig Venter, Bill Clinton, and Francis Collins. So Francis Collins was the director of the NIH Genome Centre, and that was the first one to be opened in 1991, where Jim Watson was actually the original director of that. But he stepped down, and I think it was 1993. However, another coincidence, it was published on my birthday. Not a hint for coming up soon. But anyway, we had a great party. We all got T-shirts and had champagne, and that was a good celebration. So basically you have the 2001 papers where that is called the essentially finished genome. So at that time it was the best that could be with that technology and with that time frame. So there was still work to be done. In 2003 there was a second publication where it was the finished genome, where we plugged many of those gaps. A lot of the gaps that are still in the genome. I think there was another genome published last year. But there's still a few gaps, particularly where there's repetitive sequence. So we have a lot of areas where it's just the same combination, and it's just at the moment the technology isn't good enough to get that right. And if you watch the Christmas lectures, the JVT. You may have seen this in the background. I think he talked about it in the very first episode. And this is the genomes. This is the Wellcome Trust. It's actually quite a nice place to hang out if you're waiting for a train at King's Cross. It's got really nice rooms for sitting doing some work, but also some really interesting stuff to look at. And you can actually go and read the genome, which I probably wouldn't recommend. I'd go straight to Ensemble. But each book has the genome sequence on there, which is fab. But also, this was the year that I completed my PhD thesis, so my work here was done. And on to the next stage, utilising the human genome and also genomics to understand aggressive cancers. So it's taking what I've learned to applying to new challenges. And good morning, Baltimore, is where I headed off to. So this is a bit of a tourist plug. Absolutely beautiful city, the inner harbour, the original Washington Monument, Fells Point, which is a lot of history, the original flag and things like that. They also have a street called Thames Street, spelt Thames, but the taxi driver told me it was called Thames Street. So it's pronounced how it's spelt, and he's like, that's quite right. Also the novelty of getting a yellow school bus between campuses just never wore off. And got into baseball and also a slight addiction to Old Bay seasoning for the crabs in there. So it's worth a visit if you're in that area. So not far, you're in between Philadelphia and Washington DC. There is a bit of a dark side, so you might have seen The Wire, but you just stick to the tourist paths. I can show you where they are. And yeah, I was at Johns Hopkins School of Medicine. So this is the big dome at Hopkins. So there I was working in Greg Riggins' lab on the glioblastoma kinome. So this is where I started to develop the ability to kind of build patient cohorts, so working with pathology departments, working with the clinicians about follow-up data, learning how their language and how to communicate to kind of get what I wanted and also make sure I get the right questions asked. And my main purpose was to develop that sequencing and analysis pipeline. And basically what we were doing is cherry picking the druggable bits of the genome. So basically this is just a big map really of how closely those bits of DNA and protein that I showed you earlier are related. So if they're on the same branches, they're very, very similar. If they're on different ones, then they're a little bit further apart. But they're part of a big gene family, which at the time and still is are druggable. So there were some drugs being developed that could actually target particular genes and particular pathways. So when the gene does its job correctly, there's different pathways, but if there's a mutation, that pathway can go awry and we can find a drug to basically stop it in its tracks. And this was published in PNAS and also funded by Craig Venter. You may notice his name there. So I went to the dark side, but it was all good by then. So we did the sequencing. It's two parallel projects. One with the Craig Venter Institute, who had a lot of sequences available and pipeline that we can develop. And also with the one which we did kind of in-house, kind of a smaller effort. And this formed the basis for a successful R01 grant, which is the big thing for American funding, to basically take this on and other targets to look across the whole of the kinome, but also develop the preclinical data, so looking in cell lines, but also looking in kind of mouse models to see which one we're going to go into clinical trial. And there are some that have gone into clinical trials. Also started looking at between paediatric and adults, because we hadn't really had the cohorts to do this in a lot of different diseases. So they're clinically treated differently. So you have children's wards and you have adult clinics. And the number of age is quite arbitrary. So we don't know in terms of a lot of cancers whether there is a difference, whether actually some of the adults are very similar to the paediatrics, or whether they are kind of separate diseases because of the development of those tissues. So it's also the first study that we identified a particular gene that was mutated. And also there's another name that may have spotted there. So basically Ben Carson campaigned in 2016 with the Republican nomination for president. He basically, obviously then Donald Trump take over, but that's just quite a claim to fame. He was head of the neurosurgery department at the time, so there is logic as to why he's on the paper. So then I moved back to the UK, to University of Nottingham to be part of the Children's Brain Tumour Research Centre with Richard Grundy. And basically ependymoma is a type of brain tumour that occurs most commonly in young children, but survival is still really low, and it can occur in different regions of the brain. And the treatment is still really horrible. So surgery is really the only treatment. So some of the kids have had 7 surgeries by the age of like 10. And there was very little known about it. So the work we did there was looking at particular genes, but then looking at chromosomes. So looking at a bit of the DNA, but also kind of what that DNA is doing to change the gene expression. And this was the first time I'd really looked at kind of gene expression on a large scale. And this was using a technique called serial analysis of gene expression, which I can look at my PhD student, former PhD student, they won't know, they will just know. Never heard of this one. But it basically was before its time in terms of kind of arrays and RNA-seq and the technology that I'll get onto and talk about later on. And it was a sequence-based approach for gene expression. And analysing this data, we could identify kind of what was altered and what was different between the groups. And also started to like dabble at the protein level. So this is what a lot of pathologists would look at when diagnosing different tumours. And we can use a similar technique in the blue and brown to see where expression is. And when you have something normal, this is what you should see, but actually when you have the tumour, we saw brown, which basically shows that protein is expressed. And because proteins have impact on the cell, this could be disrupting a lot of the different processes. Also part of a big study with St Jude's Children's Hospital in Memphis, where I seem to only ever go during the summer, which is, anybody's an Elvis fan, Elvis Death Week seemed to be. And I met a fantastic woman from Middlesbrough who had the keys to Elvis's teenage home, which was it's worth the good. It's just opposite St Jude, which is a strange position, but anyway, slight aside. But so this was like taking a large cohort, which actually was mostly from the UK, and looking globally and using more cutting-edge approaches than SAGE to analyse the genome. And from this, a lot of my pretty pictures do end up looking like this, with the blues and the reds. So it's basically showing each column is a different patient sample, and then each little block is a different chromosome. And within it, we've got different genes that are gained and amplified and all sorts of funny things going on. And from this, we could actually, for the first time, start to categorise these tumours, not just based on location of the brain or the spine they rose in, but actually on their biology. And part of this, Richard Grundy, the lead now, Richard Gilbertson, the leader, is now head of the Cancer Research Centre at Cambridge, doing some absolutely amazing stuff, particularly with the mouse work. Then I moved back up north to the beloved Newcastle United, but I'll just smooth over that one for now. And back to Newcastle University, where I worked with Andy Hall and Christine Harrison. With Andy, we were working on looking at paediatric leukemia. This is a paper and a collaborative effort where we were looking at specific abnormalities in leukemia and then looking at kind of amplification of a particular gene and how they then looked in mouse models to see the function of what those abnormalities meant. I also focused most of my efforts on looking at rare subtypes of leukemia. So IAMP21, this is another mouthful of intrachromosomal amplification of chromosome 21. This was a chromosome which Christine's group identified, I guess by accident. So normal screening of leukemia patient samples for a particular abnormality which they use for classifying for which treatment arm they get. They noticed something weird. And there's a few papers before this when they noticed actually it was this one chromosome where you basically had kind of this green shows kind of region of gain. So you kind of had this chromosome where you just had gains, losses, gains, and then a deletion at the end. So you had this weird, rather than just having a normal straight line, it just had lots of extra copies stacked up on top of each other. But what was the most interesting thing was it had a really, really poor outcome. And leukemia and paediatric leukemia, if you want to read about it's a really success story in cancer genomics, leukemia in the last 50 years has gone from very poor survival to kind of 99%. But there are still some subgroups that fall out where actually they're the ones that need either intensive treatment or a different one altogether. So what they did with this was I want to know what the biology was behind it. So my job was to use the technology and the data to identify kind of where there were common breakpoints. So where each tumour had a region of gain, like did they have common genes involved, and how could we look at that and try and look at the order of other abnormalities that happened to see what was going on. And we also did a project with the Sanger Centre where we really looked into the mechanism of the IAM21 and what was going on. And what the Christine's group have done is the IAM21 patients are now treated with the most intensive arm, and their survival rate is amazing. So by looking at biology of things, it's kind of how do we identify how to treat people better. But then it was moving on to my research group. So it was kind of, what do I want to work on? What's the key questions? And I attended this fantastic symposium in Dublin all about this guy called Dennis Burkitt. And basically Dennis Burkitt, he undertook a massive mission across Africa and went across 56 hospitals. And basically their mission was just really collecting really good data to understand the occurrence and distribution of lymphomas. And he identified Burkitt lymphoma, which is obviously named after him, where it's the most common childhood cancer in sub-Saharan Africa, and it still is the most common childhood cancer. It's also one of the fastest growing tumours, and down a microscope it looks like a starry sky. I'm not very good at looking at the starry sky, but I think the pathologists have got a better imagination, but it kind of does. So basically this is, each little purple dot is a tumour cell. The little stars are macrophages, so basically that's what eats dead cells. So you basically have in Burkitt's, you have a real sheet solid of tumour cells, but because there's such a high turnover of and rapid replication, you have a lot of these stars kind of munching up. But as it grows, they grow really big. They've got a lot of them on the jaw and the abdomen, but they grow really, really fast. And when I went out to Malawi to visit the ward, you could see just also with the treatment that they're given, how quickly they start shrinking as well. It's like physically over a few days, you can see that difference. It's also, in terms of cancers, it's a Rosetta Stone of cancer. So it was the first one to be associated with a virus. It was the first to be shown a translocation. So this is where two bits of chromosomes come together and either forms a new gene or forms a different way of making more copies of a particular gene. This one is MYC, so you get lots of different copies of that. And it just was a really exciting story. And also EBV is Epstein-Barr virus. I was sat next to Epstein. I didn't realise it was him until he started to do the talk, and it was just fascinating just how, again, that was an accident. I think the cells had come to the UK and hadn't got to London. They went via Manchester and something had happened, and they basically just discovered this new virus. But basically, again, bringing it back to Middlesbrough. So Blood Cancer UK, formerly Bloodwise, formerly Leukemia Lymphoma Research, formerly Leukemia Research Fund, is basically a charity that started in Middlesbrough in 1960, and this is by the family of Susan Eastwood, who was a 6-year-old who died of leukemia. And my successfully got a Senior Bennett Fellowship. You also might know of the charity through the Calendar Girls, so that was the charity for raising the money for the sofa and the family room. And it's a fantastic charity. They do all sorts of great activities as well for fundraising. So in terms of lymphomas, this is where it gets a bit more sciencey with what exciting things we're doing at the moment. So lymphomas are the third most common type of cancer in children. About 11% of cases are lymphomas. And this is basically how, WHO, World Health Organization, how they organise the different types of tumours. And there's a lot of different types of tumours. So we focus on these two. Diffuse large B-cell and Burkitt lymphoma. And they account for about 30% of the cases, but it has quite an interesting kind of relation with age. So I was saying about kind of age might be differences. So in children, Burkitt lymphoma is the most common subtype, but as you go towards teenage years, the incidence of DLBCL becomes a bit higher, Burkitt goes lower. But then when you get to adults, Burkitt lymphoma, the frequency is much less than the DLBCL. So the way that the tumours are developing is very different. It's about 80 cases per year in the UK. There's also big differences in the outcome. So Malawi, at the time of fellowship, was 50%, whereas the UK was kind of in the 90%. So in terms of the research question, what I wanted to know was, who in Malawi were under-treating, because the treatment protocols are very different. The one in the UK is very intensive. Kids are in intensive care for a long time, but as you can see, majority of them are cured, and there's not many long side effects. But in Malawi, it's a 28-day protocol, and it's very low intensity. So can we identify those ones who just need a little bit extra? In the UK, well, if we can treat them with that and it looks biologically similar, how are we over-treating? And then it's these guys who don't respond to treatment or have relapse or die from the disease. So we collected patient samples from the CCLG, which is a UK biobank. I'm going to speed up a little bit because I've realised the time. We also got samples from Malawi, from the Queen Elizabeth Central Hospital in Blantyre. This is the world's poorest country, but they had the paediatric ward open in 1997, and there's a fantastic charity led by clinicians at Newcastle, which basically built this ward and also fund a lot of the work that they do in there. So at the Queen Elizabeth Central Hospital, this is the main children's ward, actually the only children's ward. There is another half to it behind the camera, but it's very basic. And this is the science lab where the samples are taken and reviewed, and this is Liz Molyneux, who's an amazing lady, a clinician who's been working out there for many, many decades. And this data is collected in a database. So what we get in the UK are these tubes with a very, very small patch of brown at the bottom, which is actually the cells. We somehow managed to do amazing immunocytochemistry and look at the starry sky-ish appearance, look for that translocation, but also extract the DNA. And Pigeon has done some fantastic work with this and comparing the UK and the Malawi cohorts. And he's identified that FOXO1 is a particularly interesting gene which has mutations, and you can see this little hotspot here. And this changes the activity of the gene and the protein in the cell. So so going back to why we want to look at this gene, we actually found there was a gap in the data. So this is done by Alex Blaine, which was doing her undergrad project with me and now doing a PhD. We identified that actually this is the whole exon, so this we need to have a whole bit of sequence, but there's this massive hole in the middle of it. So actually the data we'd seen in the literature, well, was it right? Were we getting the right frequency? Basically we showed about using a combination of next-gen and the original Sanger sequencing, we didn't miss the data. So that work with Pigeon, we got the right answer. However, it wasn't associated with outcome, so you've got a red and a black line set on top of each other. That's the two groups with different abnormality. But he did show that there was some interesting functional analysis. This is using some cool gene editing, which he has amazingly green fingers. And you can see that there's a difference here showing a functional impact, and there's a potential for therapeutic target in the different settings. We also have identified, this is where Alex appears in terms of P53. This came out towards the end of last year, where we identified P53 as an abnormality associated with a poor outcome. And this shows how we like pretty data, I think is probably all I've got this one up for. So we like our pretty data as long as it's right. And I'll move on to this one. So identification of patient risk groups based on P53 status. So as you can see with the FOXO1, there's a clear separation of these lines. So this is fantastic because it means that for the first time in these patients, we have a molecular marker that we can go, actually, these kids need more intensive treatment or alternative treatment. But also there was a paper came out in New England Journal of Medicine at the same time where they define high-risk patients. So these are like stages of disease. So you might have heard about stage 3, stage 4 disease. And so these ones, they have clinical markers where they can do that, and they added an extra drug. So all they had the intensity, but also they added an extra drug to it. So could P53 differentiate within that high-risk group as well? And the answer is a definitive yes. So this is really exciting because even within there we can actually do P53, and the ones here in that black line, do they really need this additional drug? And that's something that we're working on now to see if we can develop a clinical trial towards. We also won a prize. So a nice cheesy lab picture from Rotterdam. Alex also won another prize at the winter meeting from CCLG. And we're also going global. So Alex has got a poster accepted for the Festival of Genomics. And I'm also on the scientific organising committee for the meeting in New York. So I'm taking Teesside to Times Square. But moving on to the NHC and the future and what we're doing. So for those of you who haven't heard the National Horizon Centre, NHC is a £22 million investment by Teesside University on the Darlington campus. And it's really a purpose-built centre of excellence for bioscience and healthcare sector, where we're bringing together research, training and partnerships. Also, coming back to Darlington, that's my granddad on the front cover. It's available on Amazon if you want to. I don't think that's exactly his uniform at that time, but unfortunately he's no longer with us to ask him the details. So bringing it home. So at Darlington we discover, develop, deliver. We also do that in Middlesbrough, so we're across both campuses, and what we're doing is discovering the diseases earlier, developing the novel treatments and delivering life-changing medicines to those in need quicker, safer and affordably, which fits at the core of all the research I do and will continue doing. So key partnerships. So when I first came here, I wanted to make sure that we were well and truly connected with all the different trusts across the region. So this is just an overview, and I'm happy to share this with you, kind of overview of the research that we're doing in this area, and the types of avenues to explore to develop new relationships and different areas of research and innovation, and also training. We also have a partnership, so this is where going back to me growing E. coli and bioreactors comes in very handy, with the local companies, so CPI and Fujifilm. So Fujifilm are manufacturing the Novavax virus, and we also have exciting things happening with CPI with RNA, their Centre of Excellence, which is almost finished. So that'll be developing kind of new types of vaccine. And here's a nice photo from our first partnership and viral vector workshop, where there's a lot of exciting projects. I also got to write "Omics is the future of bioprocessing" on a piece of paper, and it didn't come from me. Might have been some of them messaging though. Also through moving to Teesside, there's new approaches. So thinking about the patient public involvement, clinician involvement, it's a different way of thinking. I don't have to sequence anything, but kind of clinician interviews, talking to patients, talking to the public. This is through Alex's project, discussing the P53 as a biomarker and how we can translate it. And this was done through the Great North Children's Hospital with various ethical approval, and it was done through NIHR at Newcastle, and we've also got them involved in other projects since as well. So it's understanding what the impact of the work we're doing is and how we can move it forward. But going back to genomics, this is what we can do at the NHC. We've got some really cool toys. So basically my entire philosophy is here. So getting the samples, doing the diagnosis, any molecular classification, and basically just pulling the different strands apart. So there's looking at genes and then looking at clinical correlation. And we've got Illumina equipment, we've got NanoString, which is an exciting piece for looking at a lot of archival material from pathology labs, and also Nanopore, which is a really exciting technology. We also have data. So Masood is our SEO of bioinformatics and also was a postdoc with me previously. He's an absolute whiz at developing pipelines for crunching all this data in a manageable way. So previously, the Sanger has like buildings, which is where the computers are, and everybody doesn't have a building dedicated to computers. I'll try for one, but sadly we haven't got one of those. But it's taking those pipelines, it's integrating data from the literature, it's already out there, there's so much, it's taken, a lot of people have done a lot of work, but we take that to then work out how to translate it. It's also looking at different types, so looking at genes, looking at methylation, looking at DNA, and also down, when I say bulk, this is kind of the big tumour sample, whereas now we can go down to single cell and model that. Looking for genome rearrangement, so it's looking at those translocations on a bigger scale, which this data is an absolute nightmare to crunch. And then evolution, so kind of what happens at diagnosis and relapse, and which of those clones and which of those cells that aren't killed by the current treatment. And also we have Bob, so Linux, our new Linux server, Bob, and named after Bob Mortimer, bringing in the Geordie genes. Also the future, so this is a summary of kind of what I'm doing at the moment. So we have Alex as part of a project doing the care pathway. She's also going to be interviewing some other people around that, but she's looking at the microenvironment. So it's not just about that tumour mutation, it's about what else is happening in that environment, because there's a lot of, as we know, a lot of immune responses and things. We'll learn a lot about that with that C word that's been going on. We've also got some new funding for trying new non-invasive approaches. So this is taking a routine blood sample and seeing whether you can actually detect these abnormalities in the blood rather than having to go and take a big solid tumour out. And this works for other cancers as well, because if you relapse, you don't really want to go in for another biopsy if you don't have to, if you know what the tumour is. So it's trying to find an alternative way of doing it. We've also got Natalie, who started looking at aggressive bone cancers and linking this with other factors. And from filming that happened on Monday with Rare Can, so this is bringing patients, researchers and drug developers together. We've got a pilot project sequencing different samples with them. And this is one of the founders, Andy Hall, who was mentioned earlier with leukemia. Also got current studentship advertised. This is going to be led by Vasilis Lenis, one of our early career researchers, but it's bringing a new collaboration in with North Tees Hospital, who have a fantastic way of getting biopsies early. So people go into respiratory, they've got a shadow, they can get some cells, but at the moment they're not good enough for routine diagnostics to use. So we're going to be developing through genomics, but also imaging to identify, see whether we can actually detect things earlier to move them to the cancer clinics. And I've also got some new funding from NHSX working across the NHS, and also with another company and the MIC in Newcastle. And this is very different to what I've done before. It's basically looking at kind of patient adherence, so patients who go home with medicine, are they actually taking them? And this will save an absolute kind of lot of time and a lot of effort to understand how that's done. But again, it's bringing those different skill sets that we have here in Teesside together. So what is next for the future of genomics? Well, Oxford Nanopore announced this week the fastest DNA sequencing technique, 5 hours and 2 minutes. And that 2 minutes, I bet you go down to seconds. It won't be too long when they start about 2 seconds. So this is using what they call the Promethean, which is a kind of nice big stack of sequences, which is quite cool. And actually the first Guinness World Record, haven't they? Yeah. So also Nanopore have got the other end of the scale. So yes, they've got the big monster that can do the sequencing for undiagnosed patients quickly, but they've also got one that sticks on the end of your iPhone. And this is what kind of my vision for the hospital in Malawi is. They can have that on the end of their phone, they can kind of put the sample on and they can get that reading straight away. It's called the Smidge Ions. It's not available yet, but we're making good friends with Nanopore to get on their kind of early release list. There's also, you might have heard in the news about Genomics England, a 100,000 Genome Project, which David Cameron launched. But basically sequencing now is in routine. So leukemia patients and paediatric patients are sequenced. And also in the news this week, I found this via LinkedIn. There's nothing on Twitter yet about the detail. But basically X marks the $100 genome. So one of the big challenges is always about the $1,000 genome. So we've achieved that. Now it's the $100 genome. And this really just opens up so many opportunities for research in so many different areas, and also the possibility of the discovery side of it as well, just cheap, maybe super nice and cheap. So we're waiting to hear more about that. And apparently it's not just humans that you can sequence. So we've got Nick Duggett, who works on bacterial genome, we've got others working on microbiome, and he's been working with North Tees Hospital. This is one of his master's students this year, developing a real-time PCR test for basic identification for antibiotic resistance and different isolates taken from urinary tract infections. And we've also got Jamie, so I can't do a presentation without mentioning the seals. It's just I never thought I'd have conversations about demon shrimps and crabs and seals, but that basically using the same technology. So this is the Nanopore device here, which actually literally can be taken out in the field and plugged into your laptop, looking at different viruses that are invading different regions, and also diseases to advise kind of vets. So this is herpes in seals, is a bit of a problem at the moment. So some very exciting sequencing going on there. There's also other omics applications at the National Horizon Centre. We've got RNA sequencing, so this is Safwan's work, doing RNA-seq, and I will not really gauge how I understand it fully, but it's really taking that bioprocessing that I did at grown E. coli and different bugs, but it's taking those new ways of doing it to really unwrap how we reduce the costs of manufacturing and how we improve the efficiency of manufacturing in that industrial scale. And he's got some really nice funding from multiple different areas and collaborators to take this forward. We've also got mass spectrometry and proteomics. So yes, there is more than DNA, and it is the protein that does the business in the cell. So if this is wrong, then lots of things can go wrong. So this is courtesy of Claire Jennings, who is our scientific officer for mass spectrometry, looking at the different proteins, and you get various peaks, and really understanding kind of what's going on in different samples. So just to sum up, what have we learnt? The basic is omics is the past, present and the future in biosciences, healthcare, and a heck of a lot more. But you also know that I've built a career essentially doing word searches and playing spot the difference with only 4 letters, but don't tell anybody. And also that team Teesside are really leading the way, applying this latest technology to the National Horizon Centre to address the real world problems and sharing that expertise through our training and our partnerships, which is fantastic to see. And basic acknowledgements to everybody who's supported me along the way, friends, family, supervisors, mentors, the funders, and you for listening. So thank you very much for your attention, and welcome any questions. Okay, I'm fielding questions. Are there any from the audience? Steve?
You mentioned clonal evolution in terms of cancer. That feels quite a loaded phrase. What do you actually mean by clonal evolution in terms of the cancer?
So in terms of clonal evolution, we're talking about different cell populations. So we're talking about kind of basically the cell population of cells which are resistant to the treatment. So it's different from the microbiology world, but again, it is just the different cell populations and clonality. So we're looking to see what is squeezed by either the treatment or just is resistant to any treatment that we have available.
So does that mean that under those circumstances, if there is that sort of resistance to a drug, that the cancer propagates in the host, or is it still sort of an in vitro kind of?
It's basically, the cancer cells are just taking over from the normal cells. So it's taking that kind of organ and making any kind of organ or any blood sample. It's basically the cancer cells are just adapting to take over the role of the normal cells. So I mean, when you have things like radiotherapy and treatments and lose your hair and have kind of tummy upsets and things like that, it's because they are fast growing cells, and the cancer cells are faster than the normal cells. So basically it's just taking over. So it's a matter of just the ones that are in that clone that is resistant, they just spread. Yeah. And you can get those, and that's where when the blood samples come in, because we're finding more and more evidence, and there's some really cool techniques now for kind of picking up some of those cells actually in the blood and how it metastasizes and things. Safwan.
Very interesting research, particularly with regards to that Retoximab study. Now, a lot of papers have shown that monoclonal antibodies, particularly to the cancers, they work way better than these broad chemotherapy. When will doctors realize that? Because when we would actually.
So what's the question? When will doctors realise that?
When will they wake up? When we will
that monoclonal antibodies are the future of all cancer treatment. Well, we'll introduce you to some. You can spread the word. But I mean, in terms of Retoximab, so it's a CD20 basically targets, and Burkitt lymphoma and T-cell are B cells, and they have that CD20 on the surface. So that's why it's worked miraculously in adults. Diffuse large B cell lymphoma, it's been fantastic. And in the kids, it's an added thing. And basically, the trial that they published, they stopped it early because everybody was doing so well. And you couldn't have really kept that trial going with the arm without it, because it was just an advantage. However, it's like in leukemia, they've found those high-risk groups, but they've also found the ones that are lower risk to kind of de-escalate it. So there's a lot of discussions about how you would de-escalate it, because obviously if your child is cured within 6 months, would you, yes, it's horrible, yes, they're in hospital, but would you want to risk anything in the future? So a lot of discussions happening. Any other questions?
I've got a couple online, Vikki.
Okay.
What would you say is the biggest challenge of your research career so far?
Ooh, that's a very good question. What is the biggest challenge? I think the biggest challenge is funding. It's like, I think it's everything, and I think it's not necessarily the finding the right question. It's frustrating finding the right question and finding the right answer and the way to answer it. But that funding in academia and the postdoc life, it's very short contracts, and you're kind of on a contract for a year or 2 years, and it's kind of very unstabling because you can't really get your teeth into projects. This is why kind of going down that fellowship route, and also the way that Teesside kind of give early career researchers that kind of permanency It takes away a lot of that threat of, oh, in 6 months' time I'm going to get a redundancy note. So it's trying to get that has been the challenge. It's trying to think about kind of short-term and long-term at the same time.
I suppose the next one leads on from that. What would you say to a student starting out on their education in this field? Do you have any advice?
As long as you're passionate about it, because at the end of the day, you've got to convince other people that what you're doing is the most exciting thing, and if you don't find it exciting, then don't do it. Do find that strand that makes you happy. So I mean, I said microbiology, I really enjoyed microbiology, but it was actually the underlying genetics that I really enjoyed. But actually, there's a lot of commonality. So in terms of kind of how bacteria and microbes kind of replicate and kind of take over and infect and things, in terms of the cancer cell, it's kind of doing the same thing. It's basically just taking over that host environment. So just enjoy what you're doing. It will be, you have highs and lows, but just find something that you enjoy doing.
Just one more online. What developments can you see on the horizon in this field in the next 10 years that you're most excited about?
It's the technology and the costs. The fact that I think that C word has brought a lot of things to our attention, and in terms of the fact that we can do lateral flow tests at home and get reliable data is just impressive. There's quite a lot of undergrad students I've had who couldn't have achieved that during their project. Haven't had any at Teesside yet, so I'll just clarify that one. They're in the lab. So yeah, I think it's the fact that the technology and the capabilities, so we've got some projects at NHC where it is developing those lateral flow tests for other areas, but it's bringing different streams together quite quickly. And the action in terms of translating things is much speedier. Obviously, with the vaccines, everybody focused on that one problem. But I think it's just made things easier and more efficient to kind of translate research. Great. Okay. Well, thank you very much. If there's no more questions, say again, thank you again for your time.
Thanks, Vikki, and I'll just sum up. It's been a fantastic talk. Thank you very much, and really interesting to hear your career. And I think what's astonishing is how quickly that technology's moved. For me, at the beginning of my career, we were still, it was right at the beginning, and it doesn't seem that long ago, but actually it's ancient history now. And I think the way you've harnessed that technology to forge your research career is just really inspirational. So, and I think it's a shame that I'm sure many people online would have loved to have been here with you to celebrate your success. But thank you very much for an outstanding tour, and your ongoing work at the National Horizons Centre is just, again, an outstanding testament to both your ability and your inspirational leadership. So we'll end there. Thank you very much, and perhaps just another final round of applause for Vikki.